Microbiologists, we are getting ready for our second and final lab practical of the course. Just a few reminders. The lab practical is administered over 50 minutes of time through the Blackboard platform. And there is 50 questions on the practical. The practical is a combination of fill in the blank, multiple choice, and true and false style questions. The lab practicals are not cumulative, meaning that it will only contain information from weeks 8 through 13 of the course. So those are the labs that you should focus on. And the point of my brief video tutorial today is to provide you some suggestions and reminders as you begin to study for the practical. So beginning in week number 8, the first lab that you will see as part of your questions on the lab practical comes from the quantitation of cultured microorganisms lab. This was the one where you did the serial dilutions to figure out your colony forming units. And remember, CFU stands for colony forming units. And basically what we did was we used pipettes to do a series of dilutions. And again, as you're studying, make sure that you go through the background information. A lot of the bold-face terms like colony forming units are important as well. And then obviously the calculation of the CFUs, this is a very important equation. So basically what you did was you used a stock solution that consisted of soil and water. And the pipettes were used to transfer your samples for dilution. And afterwards, you were able to go through and use this formula to calculate population density. So we remember that's the CFU times the dilution factor divided by the volume of the culture that you plated. I also want to make sure that when you go through, after you look through your background information, go back through your lab. Make sure that you're familiar with what types of media you used. So for instance, this one here, we used the nutrient auger. We also utilized PBS, or phosphate buffered saline, in this lab, as well as one of the key materials. So make sure that you run back through your procedure and understand any of the specific techniques that you utilized. So your PBS was the material that was added into your tubes to create your solutions. Your soil sample made up your stock solution tube. That was your soil and water mixture. And then once you created and you did your serial dilutions, where you use the transfer pipette to transfer your sample, you then had those platings done on 6 Petri dishes. So again, just going through and understanding the key things that you've done in the lab. Materials that you've used, such as sealing your plates with that thin plastic parafilm. And remember that we always incubate our Petri dishes upside down, and that's to prevent moisture from forming on the lid of the dish and dripping onto the surface of our plates. So going back through not only the experiment itself, but also looking at the background information in the lab is incredibly important as well. So let's go back now and take a look at the next week, which is week number 9. And week number 9's lab focused on selective media and Auger. Again, going through your background information for the lab, make sure that you understand, obviously, first of all, the difference between selective media and differential media. That's really important to make sure that you understand. Remember that selective media inhibits all types of microbial growth except for the one that you're looking for. So for instance, one of the ones that we used was McConkey auger. And remember, McConkey auger was kind of a reddish purple hue to it. And it symbolized the ability to select for gram-negative bacteria that were lactose fermenters. So those were the only types of organisms that would grow on those particular Petri dishes. Differential media, we saw are types of agar that will change color. They undergo a color change when you grow a certain type of microbe on the dish. So it will allow us to see different microbes growing on the same media. So for instance, we had MSA agar, or mannitol salt agar. Remember that mannitol is a selective as an agent for Staphylococcus, and it grows the gram positives. And anytime the organism produces an acid on the plate, it turns the plates yellow in color. So acidic conditions turn mannitol salt agar yellow, and anything that's basic, it stays its normal kind of reddish color. We also talked about, so that's our differential media. I'll kind of just skip down here. So this right here is our Staphylococcus aureus, again, fermenting mannitol. It gives us a different result on the plate. The other piece that I want to bring out to you is the chemically defined media versus the complex media. Make sure you know the difference between the two. And then obviously going in and going through your 3 different lab procedures. We had the starch degrading bacteria. Remember that this was the lab that used the 0.4 starch and used iodine as a tool. We had 2 different augers that were used here. We had our nutrient auger and we also had our starch auger. And as you read through this, again, remember our 0.4% soluble starch is what was in our media. And we used iodine to tell us basically if starch was present and whether amylase was used or not. So anytime the iodine turned blue on the plate, that told us that starch was present, but amylase was not used. So make sure that you go through and you understand the techniques that we use. Any of the auger that is used in the lab is incredibly important as well. So make sure that you do read through and understand the procedures and anything that was done in the lab. We also had our selective and differential media. Make sure that you go through these. Obviously, here's our MSA auger, our mannitol salt auger, and our nutrient auger. That mannitol salt auger turned yellow in the presence of a bacteria that generated an acid. And then we also had our selection of gram negatives, which used our McConkey auger. And remember, the McConkey auger helps us to select for gram negative lactose fermenters. Bleach, again, a big piece that we see in all of our labs. Bleach is the substance that's used to destroy our Petri dishes when they're done, but also to disinfect our surfaces. All right, going into our week number 10 lab. Week number 10 dealt with the lab on differential and biochemical testing. So this was really the lab that took us into understanding our biochemical tests as well as our pH indicators. So remember that our indicators allow us to detect if a pH change has occurred to a media. So the indicator dyes are really important in telling us whether a pH change has occurred. Make sure that you go through and you understand, biochemical tests, what they are, what they're used for. And then, obviously, going through and understanding our sensitivity to antimicrobial agents. Remember that we use the Kirby-Bauer disc diffusion method, which is a radial disc diffusion method, meaning that we use antibiotic discs that were put down on the Petri dish, and those antibiotics diffused out of the discs onto the surface of the plate. And what we were able to see is a zone of inhibition or clearing that formed. With the larger the zone, the more potent the agent. So the microbe in a larger zone was more susceptible to that antimicrobial agent. If the zone was small, there was a higher level of resistance. So as you go through, make sure that you go through not only the antibiotic sensitivity lab, make sure you understand the different types of antibiotics that were used, ampicillin, kanamycin, and tetracycline. Make sure that you're also familiar with the augers again that were used, nutrient auger in this case. Remember that anytime that we inoculate our dishes, we always use the inoculating loop. The metal inoculation loop is what's used to inoculate all of our samples. And then also, we had two other tests that were done here. We had the oxidase assay, and remember that the oxidase assay is specific for the electron transport chain, or ETC, and this is a series of different intermediates that are used to transfer an electron through a series of oxidation reduction reactions, used specifically in aerobic respiration. And we see this a lot with the oxidation of glucose as one of the primary pathways that go through and utilize the ETC. The ETC for aerobic bacteria, the final step of the process, uses an intermediary that we've talked about called cytochrome C oxidase. This is the terminal complex. Any cell culture that we have that contains the cytochrome C oxidase has a reagent known as tetramethyl P phenylethylamine, and this oxidation of this substrate turns it from clear to a purple-blue color. So if the cell culture doesn't have that cytochrome C oxidase, it's not going to oxidize the substrate and it will remain clear. So knowing the science behind the labs is as important as understanding the specific types of media and things like that. We also did the catalase assay, which was looking specifically for the catalase enzyme, which allowed for bacteria to decompose hydrogen peroxide, which is highly toxic, into things like water and oxygen. And we knew that bubbles that were generated were a positive result and demonstrated the presence of catalase. So make sure you're familiar with the augers and the ingredients used, as well as the science behind the lab as well. All right, moving on. Week number 11. Week number 11 dealt with the environmental microbiology and water quality lab. So obviously this was, the key one was our Winogradsky column. Make sure that you're familiar with Winogradsky columns. You know what they are. Make sure that you're familiar with endospores. So we did a lot with the microbiology of soil and the microbiology of water quality. Make sure that you go through and you understand the labs, specifically the Winogradsky column, which use the 2-liter clear plastic soda bottle to generate your Winogradsky column. And we also utilized substances that we added in, such as newspaper. Make sure that you are familiar with the gradient that's generated. So remember, oxygen is high at the top of the column and non-existent at the bottom. And also hydrogen sulfide is very high at the bottom, but non-existent at the top. So it's allowing us to see that gradient of the aerobic and anaerobic microbes. We also talked a lot in this lab about the different environmental cycles, the carbon cycle, the nitrogen cycle, the sulfur cycle, and the phosphorus cycle. All 4 of these were prevalent in chapter 26, which is our chapter this week. And remember that carbon and nitrogen are atmospheric cycles. Sulfur and phosphorus are the sedimentary cycles. The Winogradsky column, again, environmentally had to be in a well-lit area, sunlight to help support and facilitate the growth of our aerobic photosynthesizers. And then also we were going through and doing a characterization of our soil samples, looking specifically for our endospore formers. So we utilized a endospore staining, which utilized a green stain called malachite green. And this is a differential stain, the endospore stain, which allows us to stain our endospores green. And due to the safranin as our counterstain, the vegetative or actively growing cells stained pink in color. So anytime you get this stain with pink vegetative rods and green endospores, we know that it's an endospore stain. We also obviously use our gram stain as a comparison. And the gram stain, again, uses a series of dyes as well, also differential, with the crystal violet as the primary stain, the safranin as the counter stain, and in between, we had our iodine, which is our mordant to fix the stain to both our gram-positive and gram-negative cells, and our ethyl alcohol as our decolorizer. So make sure that you're familiar with the way that you set up the lab, specifically with your candle, and using that to set up your inoculating loop and prepare your stains. All right, moving on. Our week 12 lab dealt with our food microbiology, which is the one that you have just completed, and it's our final lab of the course. So remember, with our food microbiology lab, there's a few key things that come up. We were really working with milk and yogurt in this particular lab. And specifically, we were talking about fermentation. And we saw that fermentation is really a process that generates organic acids. And make sure that you're familiar again with the background information, but specifically the assessment of the bacterial load of milk with methylene blue. Remember that methylene blue was used as an indicator of the oxygen content, so it told us how much oxygen is used up. And we also generated our yogurt. And with our yogurt preparation, remember you actually created a homemade incubator using a box lined with aluminum foil and a desk lamp as your light source. And basically what we were doing here is going through fermentation and generating a, basically using lactic acid to create our yogurt. And we were testing the pH. And remember, we allow the yogurt and milk to sit in the fridge for about a 24-hour time period once it was created. So make sure that you go through your procedure, you understand the technique that was used as well here. And with that said, that really wraps up our lab practical and the information that will be covered on the practical. So make sure that you know your augers and make sure that you know your, obviously, your background information and your lab technique. Those will be where the questions are generated from. If you have any questions, Tuesday nights, office hour, that will be the place that we talk about our lab exam. If you have questions, bring specific questions with you to go over any content that you need help with, and I'll be more than happy to walk through that. Thanks, everyone.